You know that feeling when you catch your reflection and think, huh, things are, well, changing.
Mm.
We've all been there.
Hmm.
And you might have wondered, what's really driving that process, aging, beyond just the surface level stuff?
Yeah, absolutely.
Well, you're in for a treat. We've plunged into some truly cutting-edge science that's rethinking the whole idea of getting older.
Right.
Not as just like an inevitable decline,
Oh, yeah.
but as something far more intricate and potentially reversible.
That's right. For a long time, the story we told ourselves about aging was one of, well, inevitable wear and tear, just things breaking down.
Yeah, that's what I always thought.
But what if that's not the whole picture? What if instead aging is guided by a deeper, more dynamic process, specifically at the epigenetic level?
Exactly. And for this deep dive, the material you shared was packed with fascinating research looking at the epigenome. Think of it as like the operating system on top of our DNA,
Good analogy.
focusing on things like DNA methylation and how scientists are now exploring the very real possibility of cellular rejuvenation.
It's really exciting stuff.
So for you, the listener, our mission today is to really get to grips with this idea of biological age versus the age on your driver's license. We want to uncover how groundbreaking research is investigating ways to actually, well, dial back our biological clocks.
So let's start with the basics then. You mentioned chronological age, the number of candles on your birthday cake. Easy enough.
Right.
But what does biological age actually mean for you personally?
Right. Chronological age is easy, but biological age, is that like how old your body feels?
Yeah.
Is it something like
In a
it?
way, yeah. Biological age is a much more personalized measure. It reflects the actual functional and structural condition of your body's cells and tissues at any given moment, regardless of how many years have passed since you were born. So for you, someone who's chronologically 50 could have a biological age that's younger.
Or older.
Or older, exactly, depending on various factors like lifestyle, environment, genetics.
Okay, that makes intuitive sense.
Yeah.
But how do scientists even measure something as, well, abstract as biological age? It's not like we have a little speedometer for ourselves, right?
No. That's precisely the challenge researchers faced for a long, long time. But then came the breakthrough, the discovery of the epigenetic clock.
Okay.
Think of this as, a revolutionary tool that allows us to estimate your biological age with remarkable accuracy.
An epigenetic clock.
Yeah.
That sounds like something out of science fiction. How does it even work? For you, for me.
What's truly remarkable is that scientists found they could look at the pattern of DNA methylation. That's the addition of tiny chemical tags, methyl groups, to specific spots on your DNA.
CPG sites, right?
CPG sites, yes. And they could use this pattern as an incredibly reliable marker of aging. In 2013, Stephen Horvath developed a multi-tissue age predictor based on this.
Horvath, yeah, I've heard that name.
And for you, the listener, the accuracy is just stunning. It correlates with your chronological age at about 0.96.
Wow, that's high.
It is, with a margin of error of only around 3.6 years. So essentially, your DNA holds a remarkably accurate timekeeper of your body's aging process.
Wow. So just by analyzing your DNA methylation, scientists can get a really precise estimate of your biological age. And it works across different parts of your body, like skin versus blood.
Yes, that's a crucial point for you to understand. This epigenetic clock gives consistent readings across a wide variety of your tissues, whether it's a blood sample, brain tissues, skin cells. It's not limited to just one specific area. And while Horvath's clock was a major step forward, it's worth noting for you that other epigenetic clocks have been developed since.
Okay.
And the fundamental principles we're discussing, they apply to them as well.
Okay, so this clock is ticking away in all of our tissues.
Right.
What insights does it give you, like, into the aging process itself? Is it just a number?
Well, what's fascinating for you is the ticking rate of this clock, how quickly these DNA methylation patterns actually change over time.
Oh, the speed.
Exactly. It's incredibly rapid during your early development. Then around the time you reach your 20s, it gradually slows down.
Interesting.
This rate of change also reflects an increasing diversity or heterogeneity in DNA methylation patterns among your individual cells as you age.
Heterogeneity. So, like, my cells become more different from each other epigenetically.
Exactly. It's like your cells are becoming more epigenetically distinct from one another as time goes on.
Okay.
And here's where it gets really interesting for you. Research has shown a strong link between this ticking rate and the actual pace of biological aging.
Oh, so.
For instance, studies have found that supercentenarians, people who live past 110,
Wow.
tend to have a slower rate of epigenetic aging. Their clock ticks slower, basically.
Makes sense.
On the flip side, in various diseases, your epigenetic clock appears to speed up. It's as if the clock's pace reflects the aging process itself.
What kind of diseases are we talking about here? The research had quite a list. What should I, the listener, take away from that?
Yeah, it's quite a range. For you, it's important to see the breadth of conditions associated with a faster epigenetic clock. These include things like Down syndrome and Werner syndrome.
That's the premature aging one, right?
That's the one, yes.
Yeah.
Causes rapid premature aging. We also see accelerated clocks in Huntington's disease, obesity, even some cancers.
Okay.
Furthermore, the research links accelerated epigenetic aging to things like menopause, Parkinson's disease, allergies, HIV, Alzheimer's, and even lifestyle factors like alcohol use disorder and osteoarthritis. This wide range strongly suggests that this epigenetic clock isn't just a passive marker.
It sounds like it's involved somehow.
It seems deeply intertwined with the very processes driving these conditions. Does that perhaps suggest to you that targeting this clock could have therapeutic potential down the line?
That's a powerful thought. So the fact that this epigenetic clock speeds up or slows down in sync with how quickly someone is biologically aging, whether due to like exceptional longevity or disease, it really makes you wonder if it's more than just an indicator. Could it be a driving force?
That's the crucial question for you. This strong correlation suggests, although, it doesn't definitively prove causation.
Right. Correlation isn't causation.
Exactly. But it strongly suggests that the DNA methylation clock might actually be a fundamental mechanism behind organismal aging, or at the very least, a critical component of a larger epigenetic pacemaker.
A pacemaker.
Yeah, with potentially many interconnected parts orchestrating the aging process within you.
This is truly mind-blowing for me. So aging might be this carefully orchestrated epigenetic process that naturally leads to the next big question for you. Is aging a program we're born with, or is it more about the accumulation of, wear and tear over time?
This is a fundamental debate in the scientific community, and it has direct implications for how you understand your own aging.
Right.
Are we genetically predisposed to age and decline according to some kind of preset timetable? Or is aging simply the consequence of damage building up in our cells and tissues throughout our lives, perhaps as a sort of continuation of our development?
You mentioned this term, homeoresis Waddington's moving homeostasis.
Yeah.
How does that idea help me understand this? It sounds complex.
It does sound complex, but the idea is fairly simple. Homeoresis, for you, helps illustrate that what we consider normal physiology isn't a fixed state throughout your life.
Okay.
Instead, it's more like a moving target, a moving equilibrium, constantly adapting as you develop from childhood to adulthood and beyond.
So like what's normal for a teenager isn't normal for a 60-year-old.
Precisely. Think of it this way. You wouldn't expect a car built for a long road trip to have the exact same maintenance needs throughout its entire life, right?
Yeah, of course not.
Similarly, your body's normal operation shifts throughout life, and epigenetics seems to be a key part of managing those shifts.
Okay, so this initial developmental phase from, a fertilized egg all the way to sexual maturity, for me, that definitely feels programmed. It's so precise.
Absolutely. For you, it's clear that the timing and nature of this developmental phase must be genetically programmed and, crucially, epigenetically controlled. It requires immense coordination between your different cells.
Yeah.
Now, the question for you is, what happens after you reach sexual maturity? Does this developmental program simply continue, maybe in a less organized way, eventually leading to aging?
Right.
Or does it essentially run its course? And aging, or senescence, begins when your body's repair and maintenance systems just can no longer keep pace with the ongoing damage.
Yeah.
The program ends, basically.
And there's evidence supporting both ideas, right?
Yeah.
You mentioned examples in yeasts, worms, mice, and especially plants showing signs of programmed aging. What should I take away from those examples?
For you, these examples highlight that in many organisms, aging and even death appear to be actively controlled processes, not just random decay.
Okay.
In plants, for instance, programmed cell death is a well-established part of their life cycle. And in some cases, senescence itself seems to be a genetically determined event.
But does that apply to humans?
Well, while these examples are compelling, it's important for you to remember that the extent to which human aging is strictly programmed is still debated.
Right.
Some researchers propose that aging in mammals is more of a continuation of developmental growth, perhaps driven by pathways like MTR.
MTR, yeah, that comes up a lot in aging research.
It does. It plays a key role in cell growth and metabolism throughout your life. Others suggest that biological aging is a result of a combination of these developmental and maintenance programs. And as we discussed, the epigenetic clocks might be reflecting the molecular traces of these combined processes within you.
So for me, the jury's still out. But the epigenetic clock seems to be a really important piece of this puzzle, regardless of the final answer.
Definitely seems that way.
Now, this concept of epigenetic rejuvenation for someone like me, that sounds like pure science fiction. Is it actually possible to turn back this epigenetic clock within me? Like, really?
That's precisely what researchers are actively exploring. And for you, the implications are potentially transformative. The idea of epigenetic rejuvenation is all about attempting to reverse those age-related changes in your epigenome.
Making the clock tick backward.
Essentially, yes. Making your epigenetic clock tick backward towards a younger setting.
Where did this radical idea even come from for me to consider? It seems so out there.
Well, think about embryonic stem cells, ES cells. Biologically, they represent the youngest possible state.
The ultimate blank slates.
Exactly. They have the remarkable ability to divide indefinitely and essentially remain in a state of suspended animation. Interestingly for you, it's important to know that in ES cells, the epigenetic clock is silent.
It hasn't started ticking yet.
Correct. And even your circadian clock, your internal daily rhythm, is inactive until these cells begin to specialize into specific cell types, differentiate.
So for me, the clock starts ticking when a cell decides on its specific job and identity within my body, like becoming a skin cell or a neuron.
Exactly. And for you, the first real indications that we might be able to rejuvenate your somatic cells, your regular body cells, came with the development of animal cloning back in the 1960s.
Dolly the sheep and all that.
Right. And then much more recently with the groundbreaking work on cell reprogramming by Takahashi and Yamanaka.
Ah, Yamanaka factors, right.
Yeah.
That famous cocktail of genes that can essentially rewind an adult cell back to a stem cell-like state. For me, that still sounds like actual magic.
It feels like it sometimes, yes. Their work demonstrated that your adult somatic cells could indeed be reprogrammed to become induced pluripotent stem cells, or IPS cells.
IPS cells.
What's significant for you is that this reprogramming process halts the epigenetic clock and resets the epigenetic age of the cell to almost zero.
Zero, wow.
Making them very similar to the cells found in a very early embryo, a blastocyst. It's as if the cell's epigenetic history has been completely erased.
That's incredible to think about for me. So you can take an old skin cell from someone, reprogram it, and it becomes epigenetically young again.
Right.
Really young.
That's right. And there have been some truly compelling studies demonstrating this very phenomenon. For instance, a French research group led by J.M. Lemaitre took skin fibroblasts, skin cells, from very old, healthy centenarians.
People over 100.
Yes. And reprogrammed them into IPS cells using a specific cocktail of 6 reprogramming genes. Then, and this is key, they guided these IPS cells to differentiate back into fibroblasts.
Okay, so they made them young stem cells, then made them skin cells again.
Exactly. And here's the amazing part for you. These newly generated fibroblasts exhibited characteristics of young cells.
Like what?
Their gene activity patterns, the function of their mitochondria, the cell's powerhouses, even the length of their telomeres.
Telomeres, the gaps on chromosome.
Right. They all resembled those of cells taken from young individuals. The researchers concluded that the centenarians' fibroblasts had been effectively rejuvenated.
That's a really powerful demonstration for me that the underlying software, the epigenetic information, might still be largely intact even in very old cells. It's not totally corrupted.
Exactly. You used a fascinating analogy earlier, the interplanetary probe one. Can you explain how that helps me understand this better?
Oh, right, the Alex Comfort analogy. It helps you visualize the idea. Imagine a probe sent on a long mission to Mars.
Okay.
It has an onboard computer constantly monitoring all its systems. Even if individual parts experience wear and tear over the long journey, maybe a solar panel gets dusty or a sensor degrades.
Right. Stuff happens.
But if the core computer and its software remain functional, mission control back on Earth can send new commands. Maybe commands to refuel and reignite the engines for a return trip.
Okay.
If the probe responds successfully, it suggests that the crucial components and the information within the computer were preserved, despite the harsh conditions and the passage of time.
Ah, I see.
Similarly, these cell rejuvenation studies suggest to you that even in your aged cells, the fundamental epigenetic mechanisms that control cell function remain largely responsive. They can still receive and execute commands, like the ones from the Yamanaka factors.
That's a really helpful way for me to think about it. It suggests that aging isn't necessarily about irreversible damage to the fundamental information, the blueprint.
Seems that way.
But you also mentioned an alternative idea about epigenetic disorganization. What should I know about that? Is it just wear and tear again?
Well, yes, some more recent research in mice, known as the ICE mice studies.
ICE mice.
Yeah, it stands for Inducible Changes to the Epigenome. This research proposes to you that aging might be due to a gradual disarray of the epigenome, like things getting messy,
Okay.
and maybe a loss of crucial epigenetic information resulting from DNA damage, particularly DNA breaks, rather than just the accumulation of genetic mutations.
So breaks in the DNA disrupt the epigenetic organization.
That's the idea here. These breaks disrupt the carefully organized epigenetic landscape within your cells. The researchers even speculated about a potential backup copy of epigenetic information.
A backup copy, how'd that work?
Well, that's the question. To explain how aged cells can still be reprogrammed if the main copy is disorganized. But this backup idea raises questions about its evolutionary purpose, Since significant natural rejuvenation doesn't really occur in mammals.
Right.
So, applying Occam's razor, the principle of favoring the simplest explanation.
Keep it simple.
The more straightforward idea for you is that the epigenome remains largely intact even at advanced ages, which is why it can be reset through reprogramming. The main copy is still good enough.
Okay, that makes sense. So full reprogramming seems to work in the lab, resetting the clock and rejuvenating cells. But as you said, it can't just be used directly in a living organism like me because of the risk of teratomas, these uncontrolled cell growth, tumors, essentially.
Right, that's the big hurdle.
So what's the potential way forward for us? How do we get the benefits without the risks?
This is where the concept of cyclic partial cell reprogramming comes into play for you. It's a really clever idea.
Partial reprogramming.
Yeah.
Okay.
Instead of fully reprogramming cells all the way back to a pluripotent stem cell state, this approach involves using multiple short bursts of activating and then deactivating the reprogramming genes, like pulses.
Short bursts. Why?
The goal here is to erase some of the epigenetic marks of aging, turn back the clock a bit, while still allowing the cell to maintain its specific identity and function within your body. Keep it a skin cell or whatever it is.
So for me, it's like hitting the rewind button on the aging clock, but only partially, and doing it in controlled pulses so it doesn't go too far.
Precisely. This strategy aims to rejuvenate your cells without causing them to completely lose their specialized roles, and crucially, without risking forming tumors.
Okay. Has this been tested?
Yes, we know of a couple of documented in vivo studies in living animals using this approach that you should be aware of.
Tell me about them.
One study was in progeric mice, those mice with accelerated aging. They found that cyclic partial reprogramming significantly extended their lifespan and partially rejuvenated some of their tissues.
Partially rejuvenated.
Yeah, it didn't completely reverse all signs of aging, but it made a difference. Another study looked at middle-aged mice, focusing on the hippocampus.
The memory center in the brain.
Right. They found that cyclic partial reprogramming partially reversed the age-related decline in a specific histone modification, another type of epigenetic mark, and even improved the mice's performance in a memory test.
That sounds promising for us, especially the memory part. But you mentioned earlier that epigenetic age wasn't directly measured in these partial reprogramming studies.
That's correct. That's a key limitation. So while we observed some encouraging changes in the animal's physical characteristics and functions, we don't know for sure the precise extent to which the epigenetic clock itself was reset in those specific studies.
Okay, so we see some benefits, but we don't know exactly how much the clock moved.
Right. To better understand the potential outcomes of partial reprogramming for you, researchers have proposed a model of the epigenome with different modules. It helps conceptualize it.
Modules, like different parts.
Yeah. Imagine your epigenome has at least two main parts. Module A, which represents the DNA methylation clock we've been discussing,
The clock itself.
and Module B, which encompasses the rest of the epigenome, including all the epigenetic marks that define what type of cell it is, a skin cell, a brain cell, etc., its identity.
Okay, so clock module A and cell identity module B.
Got it. Okay, so if you do full reprogramming, like with iPS cells, you essentially wipe clean both Module A and Module B, taking an old cell all the way back to a blank slate.
Right. Total reset.
What happens with partial reprogramming in this model, and what are the implications for me? What could happen?
Well, there are a few possibilities for you to consider. One scenario, the ideal one maybe, is that partial reprogramming could erase age-related marks from both Module A, the clock, and Module B, other age marks, while fully preserving the crucial cell identity marks in Module B.
So younger clock, younger function, same cell type.
If this happens, yes, we might see a more comprehensive rejuvenation of the cell's function and characteristics. That would be great. However, another possibility, maybe more likely based on current data, is that partial reprogramming might primarily target Module A.
Just the clock.
It might reset the epigenetic clock to a younger reading, but leave some of the other age-related changes in Module B relatively untouched.
So what would that look like?
In this case, you might see a young epigenetic clock reading, which is good, but only a partial improvement in the cell's overall function and appearance. The cell looks young by the clock, but maybe doesn't function fully young yet.
And the studies so far suggest?
The initial results from those in vivo partial reprogramming studies seem to suggest this latter scenario, resetting the clock, but only getting partial functional rejuvenation, is maybe more likely, at least with the current methods.
It makes sense to me that it might be more complex than just rewinding a single clock. It's probably interconnected. And you made a really important point earlier that these OSKM genes, the Yamanaka factors, probably didn't evolve in adult organisms like us to combat aging naturally.
Exactly. That's key. Their primary role appears to be in the very, very early stages of development, specifically in resetting the epigenetic age to 0 in the zygote.
Fertilized egg.
Right. Setting the clock back to the beginning for the new organism. It's unlikely they were intended by evolution to be continuously active in adult tissues like yours or mine.
This leads us to the contrasting effects of Yamanaka genes depending on how they're delivered. This was a really surprising finding in the research for me. It seems contradictory.
It is quite remarkable and a bit confusing at first glance. It turns out that continuously expressing the Yamanaka genes within a living organism can lead to very different outcomes depending on how they are introduced.
How so?
When delivered using certain viral vectors in a targeted way, they've demonstrated some truly remarkable regenerative effects that could be relevant to you.
Like what?
For example, one study showed that delivering OSK genes, just 3 of the 4 Yamanaka factors, directly into the eye using a specific type of safe viral vector, AAV2, was able to reverse vision loss in mouse models of glaucoma and age-related vision impairment.
Wow. Reverse vision loss.
Yes. And importantly, all without causing any apparent harmful side effects or uncontrolled cell growth like teratomas in the retina. That suggests the possibility of very specific, localized rejuvenation without dedifferentiation.
That's incredible to think about for me. Reversing vision loss through gene therapy like that. That's huge. But you also mentioned that expressing these same genes throughout the bodies of transgenic mice had some really negative consequences. What should I know about that contrast? It seems weird.
Yes, it's a stark contrast. When the Yamanaka genes were induced to be expressed systemically throughout the entire bodies of genetically modified mice.
Not targeted delivery.
Exactly, just turned on everywhere. It led to rapid weight loss, severe tissue abnormalities, particularly in the digestive system, and ultimately rapid death. This really highlights for you that the context and method of delivery are absolutely critical when it comes to manipulating these powerful reprogramming factors. Systemic continuous expression appears to be quite toxic. Targeted, maybe vector-mediated, seems potentially safer and regenerative.
So for me, it's all about where and how these genes are activated. Location, 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 and delivery method. What about in human cells directly? What kind of effects have been observed there that I should be aware of?
Yeah, there are some interesting human cell studies, too. Studies using human umbilical cord cells showed that long-term expression of the full OSKM cocktail using a different type of viral vector, an adenoviral vector, didn't cause toxicity or uncontrolled cell growth, surprisingly, but it did cause the cells to kind of reorganize within the culture dish. Cellular redistribution, they called it.
Not tumors, but changes.
Right. Furthermore, transient delivery, very short term, of a slightly different combination of reprogramming messenger RNAs, using mRNA instead of viruses.
mRNA, like the vaccines.
Exactly. Including factors like Lin28 and NANOG, in addition to OSKM, so OSKMLN. This was able to rapidly reverse markers of cellular aging in human fibroblasts and endothelial cells without changing their fundamental cell identity.
So rejuvenation without reprogramming.
With transient delivery, yes. However, if this mRNA cocktail was expressed for a longer duration, it did lead to full reprogramming into iPS cells.
Ah, so duration matters too.
Absolutely. This really underscores for you how the duration and method of Yamanaka factor expression can steer the outcome either towards rejuvenation or full-blown reprogramming. And as we touched on, viral vector delivery might somehow prevent this complete loss of cell identity, this dedifferentiation, even with continuous gene expression in certain contexts like that eye study. It's complex.
This is fascinating for me. It's like we're learning the rules of how to use these powerful tools safely. Now, moving beyond directly manipulating gene expression with reprogramming factors, what other non-reprogramming strategies are being explored that I should know about? Are there other ways to potentially turn back the clock?
Yes, definitely. There are some truly intriguing non-reprogramming rejuvenation strategies being investigated that could have implications for you. Things that don't involve Yamanaka factors at all.
Okay.
One notable example is the TRIME trial in humans. TRIME stands for thymus regeneration, immunorestoration, and insulin mitigation.
Tell me more about that. The TRIME trial sounded particularly interesting because it was in people.
Right. The TRIME trial involved a small group of healthy men between 51 and 65 years old. They received a combination therapy of growth hormone, metformin, and DHEA for 1 year.
Okay, those are known substances.
Exactly. Growth hormone, a common diabetes drug metformin, and a hormone precursor, DHEA. The results were quite surprising for you. They observed an average reversal of epigenetic age, measured by several clocks, by about 1.5 years over the 1 year of treatment.
So they got biologically younger while getting chronologically older.
According to the epigenetic clocks, yes. And this was accompanied by improvements in various measures of immune system function. The trial was actually initially designed to regenerate the thymus gland.
The immune system organ.
Right, which is vital for immune health and tends to shrink and decline with age. The epigenetic reversal was almost a secondary finding.
So for me, that's an example of a combination of existing, well-known substances potentially having an anti-aging effect on the epigenome.
Without reprogramming genes.
Exactly. It suggests other pathways can influence the clock. And then there was this preliminary study in rats using a protein-rich fraction derived from the blood plasma of young rats. They even referred to it as an elixir in the research paper. What did they find that's relevant to you?
An elixir. That sounds very intriguing. What did they discover that I should know? Was it like young blood transfusions?
Sort of, but it was a specific protein fraction, not whole plasma. They found that repeatedly administering this young plasma fraction to older rats over a period of 5 months led to a significant reversal of epigenetic age in several of their tissues, liver, blood, and heart.
Significant reversal. Yes, their epigenetic age shifted measurably closer to that of young adult rats. And importantly, these epigenetic changes were also accompanied by functional improvements in various blood and biochemical markers. Things got healthier.
But was it a complete reversal?
No. And it's important for you to note that the old rats weren't completely returned to a youthful state. For instance, their body weight, which typically increases with age in these male rats, wasn't reduced by the treatment. So some things changed, others didn't.
So similar to what we maybe saw with partial reprogramming, it seems like a significant epigenetic reset, but maybe not a complete return to a fully young state for the organism. Some aspects of aging seem resistant.
That seems to be the pattern so far.
What's the current thinking on why that might be? Why only partial reversal?
That's the current puzzle. And for you, the researchers in the rat plasma study even proposed a trimodular model of the epigenome to try and explain their findings, adding another layer.
Trimodular. So 3 parts now.
Yeah. In this model, module A is still the epigenetic clock. Module B represents parts of the epigenome that are responsive to this elixir from young plasma. And module C represents the portion that appears to be unaffected by it, the resistant part.
Okay, clock, elixir responsive, elixir insensitive.
Something like that. This suggests that different rejuvenation strategies, reprogramming, drugs like in TRIME, young plasma factors, might target different aspects, different modules of the aging process at the epigenetic level. No single magic bullet hits everything, perhaps.
It really feels like we're just beginning to scratch the surface of this incredibly complex process of aging, and it's clear for me that the epigenome is right at the very center of it all. It's the key intersection.
Absolutely. We've come such a long way from simply accepting aging as an inevitable, irreversible decline. The groundbreaking discoveries of cell reprogramming and the epigenetic clock have truly ushered in a new era.
Yeah.
An era where we're starting to understand aging as a potentially modifiable epigenetic process that directly impacts you, your health, your lifespan.
And the strong correlation between how the epigenetic clock behaves, speeding up, slowing down, reversing, and the actual rate of biological aging strongly suggests to me that it plays a fundamental role. Maybe not the only role, but a key one, perhaps even as the central timekeeper or part of it.
Precisely. And the fact that we can now apparently make this clock tick backward through various epigenetic rejuvenation strategies is a potential game changer
Huge.
for both understanding the fundamental mechanisms of aging and potentially developing future interventions that could impact your health span, the years you live in good health.
It's still early days for in vivo rejuvenation research, as you've highlighted throughout. Very early. But these initial results showing partial improvements in health and function when the epigenetic clock is reversed, even partially, they're incredibly encouraging for me to hear. It feels like progress.
They are. It's cautious optimism, but it's there. Epigenetic rejuvenation, whether achieved through partial reprogramming techniques or these non-reprogramming approaches like drugs or plasma factors, really holds the key, I think.
The key to what?
To both unraveling the core mechanisms of aging, figuring out why we age at the molecular level, and developing future therapies that could benefit you directly. Real treatments.
So, considering all of this fascinating evidence, it really makes me wonder, what does the future of aging interventions might look like? What's the most likely path forward? Will we be targeting specific epigenetic marks with drugs?
Could be. Precision epigenetic editing.
Will partial reprogramming become a viable therapeutic approach for humans safely?
That's the big question for reprogramming, safety and efficacy.
Or will we be harnessing factors found in young organisms, like that intriguing elixir from the rat study, trying to bottle youth?
Also a very active area of research, identifying those specific factors.
And then, of course, looming over all of this, there are the broader ethical and societal implications of potentially being able to significantly slow or even reverse biological age. It's a lot for me to think about. Who gets access? What does it mean for society?
It certainly is. These are the vital questions that will drive future research, not just the science, but the ethics and policy around it in this rapidly advancing field. And ultimately, they'll shape our understanding of what it means to age for all of us.
Well, this has been an absolutely fascinating deep dive into the science of aging and the, well, really exciting possibilities of rejuvenation. Thank you so much for breaking down all this complex information for us. My head is spinning a bit.
My pleasure. It's a constantly evolving field, and there are always new and intriguing discoveries being made that could ultimately impact your life, my life, everyone's life.
For those of you listening, we sincerely hope this deep dive has provided you with a clearer understanding of biological age, the crucial role of the epigenetic clock, and the truly groundbreaking potential of epigenetic rejuvenation.
Hope it was useful.
We encourage you to really ponder the questions we've raised at the end there about the future, the ethics, and perhaps even explore some of the fascinating research we've discussed, if you're curious. As always, we'd love to hear your feedback and any suggestions you might have for future deep dives.